Gary, for the introduction, and thank you to the organizers for inviting me. I'm going to just get the screen set up here. So what I'm going to talk about today is really a focus on a small molecule focus centered approach to thinking about the dark kinome, and by implication, the light kinome as well. And it derives from the work that we've been doing as part of the illuminating the function of the understudied kinome as part of this Common Fund program. So a couple of the questions that I'm going to address today is this question of which kinases are inhibited by existing chemical ligands, and particularly those that have been developed all the way to therapeutic drugs. How do we reconcile different measures of target binding affinity, particularly the routine assays that are used in drug discovery, and I'll show those in a moment. One of the lessons that's coming out of the use of these target binding methods is that there are, even for what we think of as the most selective kinase inhibitors, small molecule inhibitors, with rare exception, they actually inhibit multiple kinases. And those of you who know the history of their development in oncology will know that in many cases, the secondary targets actually turn out to be part of the therapeutic mechanism. And there are even cases such as crizotinib, for example, that starts off being a target for one kinase, in that case, the c-Met inhibitor, and ends up being primarily clinically useful in a completely different disease for a different kinase, in that case, the ALK inhibitor in lung cancer. So this polypharmacology is true of even the most highly evolved compounds. And there is an argument that I won't try to sustain today, but that's coming out through some of our work with Novartis, that in fact, drugs with slightly more polypharmacology may, on the whole, be more efficacious. And more surprisingly, and this we really don't yet understand, they may actually be less toxic in some cases. So can we take what is observed really in points 1 and 2 and begin to actually make this a predictive feature of drug discovery using either conventional methods or what Jarrod's going to talk about later with fragments? And then finally, can we actually use just measurements and some amount of theory to actually begin to probe dark kinases? And the answer is, we think yes, but I'll just show you a couple of examples where approved clinical grade compounds actually have as their highest affinity targets dark kinases that have hitherto been relatively little studied. So the way we're trying to actually get at target deconvolution is actually threefold. One of them is to do direct measurements. And we do two types of measurements. One are multiplexed inhibitor bead assays. And these are assays in which an ATP-like moiety or a drug-like moiety is attached to a bead. Those are then incubated with extract, and you then pull down and get all the bound kinases and other ATPases, and then you do that in the presence and absence of a competitor drug. And the competition tells you which, in principle, tells you which are the selected hits for that compound. And that's an assay that's been available for over 10 years. And as part of this effort that Gary leads, we've been trying to develop this into a multiplex TMT-based assay that is also targeted, so targets the entire kinome. And the goal there is to make it more precise and more efficient. The second thing we did, and I'll show you the data for this, is some large-scale kinome scan assays. These are actually done, this is a fairly classically done assay in companies, because we were sort of shut down for a while during COVID. It was an efficient way to actually use our M&S resources in this grant. In this case, what's done is fragments of kinases are actually expressed, and then a competitive binding assay is done, and then they're sequenced directly. And so the kinases are made in recombinant form, and they contain sequenceable tags. We can then look at biological activity, and that can be done either with the literature or directly in cell lines. I'll allude to that briefly. And then finally, the largest and growing effort is to use modern AI type machine learning tools to actually improve structure prediction and computational docking. And so docking has been around for many years, but those of you who followed AlphaFold and similar methods know that there's been a big improvement in the accuracy of these methods for structure prediction within the last year or two. And the question is, can we extend that now to computational docking? So there are two efforts as part of the dark kinase, one that's coming out of our group and with Nathaniel Gray, and the other one out of Tim Wilson, both of which are trying to build optimized kinase libraries. And those are largely focused on trying to annotate the very extensive literature in here. And there is a website that we have available through the Dark Kinase knowledge base. And here's an example of a publication of this, in which you can go in and put any given target in. Here was a BRAF, for example, and then it'll tell you what are the most selective compounds on there. And it's designed to try to find the compounds with the maximum selectivity, which are maximally divergent in chemical space. And this could be made even more powerful if we could make sure that in off-target space, they were maximally divergent. So if your biological question focused on BRAF, you would have 2 or more compounds that you could use, and you could then do a sort of kind of simple structure type activity assay to make sure that in fact the biology you were focusing on was primarily that kinase, even though any single probe would inhibit multiple enzymes. So the library that I'm going to show you about today is one that we put together. It's the second round of the IDG library. And this gives you a sense of the selectivity classes. I'll refer you back to that paper, what we mean by selective. But to the first order of approximation, most selective kinase inhibitors hit between 2 and 5 targets at similar affinity, semi-selective up to 10 to 15, polyselective or broader. And then there are others which are less known. And we also put them in this library together to have all approved clinical compounds, regardless of the selectivity, because those were felt to be of fundamental interest. And the idea was to make the library as small as possible, around 350 compounds. And we're now, it's publicly available. We're now working with vendors to make it commercially available. And this would be the substrate for doing reasonably efficient small screens, or targeted screens, I should say, on the dark and light kinome. Now, the Kinome scan assay that I alluded to before is the one that many of you will have seen. Here's sort of on the left, this immobilized ligand, the DNA tagged kinase, done with and without competition again. And then this is all done within a company. Eurofins has worked with us over the last couple of months to do this in what I think is the largest public domain effort, so all the data to be released. And it gives rise to those very familiar sort of circular kinome trees you see on the right, that are in many publications, where the size of the dot gives you a sense of the degree of the selectivity, or the fraction in this case of inhibition. So here's an example where what we've now done is taken a whole kinome screen for them, so you can run these assays for single kinases or the whole kinome, and it's about 90% of what we would think of as the annotated kinome, including dark kinome. It has to do with technical reasons that they haven't expressed the others. We ran that against the Kinome scan library, and then this was done now at 100 nanomolar. And you can see, and the degree of inhibition is indicated by the darkness of the heat map. As you can see immediately that, there are areas of the kinome that are poorly covered, some that are relatively well covered. You can see there are whole areas where there's blocks of kinases that seem to be broadly inhibited by all compounds. That's been known anecdotally in the literature. We're trying to understand why that is, but you'll see that this wouldn't correspond to having one kinase inhibitor for each kinase. It's really much more of the sort of kind of blocks that you see of dark red here. And here's now run at 1 micromolar, which if you do a single shot assay is the recommended concentration for this assay in the company. Precisely why that's recommended, we don't yet know, but this is what they would say would most commonly be done. And I think it emphasizes what I showed in the previous picture, which is that actually many compounds, including many clinical grade compounds, actually hit large fractions of the kinome to different degrees. The question from a biologist point of view, of course, would be, is this adventitious, or is it in fact functional and significant from a mechanistic standpoint? And secondly, from a chemical standpoint, is, how could we make use of this information to design either more selective inhibitors or to fill in some of the areas shown yellow here where there's relatively little chemical matter. So here's sort of a co-clustering of that information, where we simply co-clustered based on, and we're putting the Gini index here, and we're clustering both on inhibitor and kinase. So it's the same information you've seen before, but now with a two-way clustering, just to emphasize the subset of the kinases on the upper right, which have many compounds affecting them, the subset on the left. And what's interesting, if you look at what we're calling here the light kinome, so those things excluded from the druggable genome project, around 380 kinases, you'll see that actually it's pretty similar visually to what you see with the roughly 160 kinases that live in the dark kinome. There's a statistically significant difference here, more coverage of the light kinome than the dark, but the size of the effect is really, really very small. So that tells us that existing chemical matter very substantially already covers the dark kinome, although those would be, in many cases, compounds that are polyselective, but that shouldn't be seen as different from what we see with the light kinome. And here now is a sort of subset of that, and this is just a couple of these so-called dark kinases. They're indicated with the gray circles here, and we've actually now done ex vivo activity assays. So these are actually ATP phosphorylation activity assays done in vitro, and we've done that across the nominal targets. So lastartanib nominally is an inhibitor of FLT3, for example, and you can see that one of the understudied kinases, MAP3K19, is in fact a higher affinity target. And you can see that across the whole set of them. This indicates also a clinical phase. Lastartanib, I think actually is now approved, but you can see sunitinib and sorafenib, for example, very widely used, have a series of targets that are really among their highest affinity targets of all. And then down at the bottom are cases where it's clear that the light kinases, the nominal kinases, are probably the primary targets. So interesting spectrum, not trivial to work out from this, whether, for example, with lastartanib, MAP3K19 is in fact part of the mechanism of action, and that's likely to be tumor type. But that would be the next phase of this analysis. So how many kinases are inhibited then per compound given this optimized library? And so we can score this at different thresholds. I'll just actually put both of them. So less than 1% activity and less than 10% activity. You can see there's obviously some fraction that's really poorly covered at the very highest selectivity. So activity is in the rows here, and the columns are the concentration of the drug. But you can see that on the whole, the total number at this sort of reasonably good coverage, the 1 micromolar number that I mentioned before, you can see that typically the average is around 10 kinases inhibited to similar degrees by all compounds in the set. And so that's sort of become our sort of working rule of thumb, with some compounds being as few as 2 targets and some like sorafenib having well over 100. So this is the area that we've really started to focus on, sort of this middle domain where you have some degree of coverage with some degree of selectivity. And so the other way to get an assay of this that is really accessible to us in the lab now that we're all back in operation is to use chemoproteomics, and these are the multiplexed inhibitor bead assays that have been developed by several labs, notably Gary Johnson's as well. And in this case, you'll use some sort of labeling. This is actually a light heavy labeling shown here, but we actually do this with TMT to give us an 18-plex pool. Once again, there's a cell lysis. You bond to beads that carry drug-like moieties on them. You do pull-down assays, and then you do that in the presence of competition, and from that you infer the selectivity. And the advance that we've been making and are really almost ready to go full-time now is to do the TMT, as I mentioned, 18 parallel assays, so you can do direct comparison and dose response, but to do them with labeled peptides. So the whole thing is done as targeted proteomics. And here's just an example that you can look at where we take a compound that had been of interest in the lab in one of our dark kinase assays, KW2449, and we can see that the effect size and the p-value as we bring in competition at different concentrations of this drug, and you can see the primary targets coming out. And there are kinases in there, but of course there are other proteins as well. And in some cases, the therapeutic significance, I would say, of these other proteins is unknown. Most of them are, most of the selectively released ones in such an assay are also ATPases. So here's sort of the concept behind the 16-plex TMT, or now 18, that we can do. The idea is that you can do benchtop fractionation with multiplex inhibitor beads. That's fairly straightforward. But the goal was actually now to put in these trigger peptides, these labeled peptides, and the method is called Tomato from the Gigi lab. There's no fractionation at all, so you skip the first step, you take the entire mixture, and we can get dose response curves from 18 compounds in about 3 hours. And that's now been demonstrated for a fraction of the kinome by Marion, Gary, and Robert, the names listed at the lower right. and is now being extended to the entire dark and light kinome. Given the previous evidence that made no sense to look only at dark, we may as well put that in the context of the kinome as a whole. And this shows just an example of that working. We've been getting peptides in the last year has been not so easy. We were hugely helped by Reed Townsend, who's also a member of the project. He gave us an initial set, and we've now built a total of 1,040 peptides. It's conceivable these could be made commercially available, but certainly the sequences will all be available, and then we've labeled them with TNT. Now, one of the questions is, given that we can sort of measure and enumerate this polypharmacology, the question is, can we actually understand it in any reasonable way? And so here's a representation of the polypharmacology using Bayesian affinity fingerprinting. And so the idea is to take the kinome tree, which you may recognize on the right. It's the classical sort of depiction of the kinome based on sequence similarity at a protein level. And then we're doing a two-dimensional clustering, and the Bayesian affinity clustering is designed to look at compounds with similar chemical structure. So think of it as a compound-centric probe of the ATP binding pocket that would be, at least to some extent, predictive of polypharmacology. It's not yet predictive, it more captures the polypharmacology. And then we can take those clusters here and we can map them onto the kinome tree. And what you can see is that the different clusters don't map onto the phylogeny of the tree in any simple way. That is to say that kinases that are close together, not atypically, will be inhibited by the same molecule, but in many cases, that's no more true than of compounds that live elsewhere in the tree. So the genomics in this case does not guide us in a useful way to understanding the polypharmacology of these compounds. So we've also been now trying to work out other ways of doing this on a functional level. And here we've been trying to use the Blode's connectivity map. So could we relate compounds together in function space and then in structure space? And those will be the last two topics I show you about. So to get at functional space, we wanted to make use of the connectivity map that's at clue.io and is the product of the LINKS program, an earlier program from the Common Fund. And what happens there is that in the connectivity map is you have a large number of, I think it's now up to 2 million examples of perturbed cells, either with small molecules or with RNA knockout, and then a transcriptional profile is done. And you can then query that database either with a compound or with a transcriptional profile, and then you can ask to what extent are those things similar to each other. And so we're now trying to add the connectivity map to what I showed you here, this sort of sequence-dependent clustering of kinases, giving us the classical map, a structure-based approach that I'll describe in a minute, giving us insight into the ATP binding pocket, and then the binding data. And then the idea is that what will come out of the project, focused for this program very much on the dark kinase, of course, is sort of a proposed map that can be looked at in several different ways. And ultimately, we'd like an integrated look. How does it look from structure? How does it look from genomics? How does it look from function, the Brode connectivity map? And how does it look from predicted or measured ATP binding pocket? Now, one of the issues, of course, in all of this is that the structural conservation of these kinases is not captured by sequence similarity in any way, and that goes well beyond the ATP binding pocket. And this is now classic work that many of you will be familiar with, trying to line these things up and build them into these phylogenetic trees. And so what we've been doing is taking all of the kinases for which there are structures, bringing them into a uniform alignment so that you can actually get all the PDB structures, and take all the missing structures and then predict them with AlphaFold, and then take the measured data that I showed you from Eurofins here. We can actually get a binding score out of that, and then we can do docking in a modified form of AlphaFold to actually get the affinity in kcals per mole, or the binding energy in kcals per mole. So here's a sort of classic docking on the left that we've done with a structure that's been predicted by AlphaFold, and on the right is another example of a structure which we've done that came directly from PDB. And so these are anecdotes at the moment. We don't yet have large-scale energetics that I can share with you, but this looks very, very promising, and we're imagining that we will take all of the library I showed you against all of the kinome and have dark structures with affinities available by a little bit later in this year. And then those will be the substrate on which a learning engine will be put, machine learning engine, to try to make this polypharmacology both more interpretable and more predictable. And here's sort of where that sort of lives. I want to make sure I don't take up too much time here, but the idea is to take, here's sort of the first paper that we're envisioning here, is to take a subset of these kinases, leaving aside the receptor tyrosine kinases here. And you try to build very, very long sort of descriptor maps of those on which you can perform conventional learning. And the idea of those descriptor maps is that they try to pull out the features of the active site preferentially. And those would be include things like solvent exposed area, et cetera. And those can all be calculated now from these structures. And then eventually those get compressed. And then the idea is that those result in this sort of actionable description of the kinase. So I know this is fairly superficial, but if anyone's interested, please write to me. It's sort of work in progress, trying to combine structure work with machine learning, something that we talked about in a paper at the end of last year in Nature Methods. So I think one of the questions that has arisen as we've gone into this library is how well can we take resources that are available, like the connectivity map that I alluded to before, and actually query them with newly derived signatures, because the structures that I showed you and the compounds are not all in the existing resource. And that has been one limitation of that resource. It's based on a technology called L1000 profiling that's not broadly available to us. And really not to anybody. And so here's an attempt to ask, can we do high throughput conventional RNA sequencing for less than $10 a well is the goal against compounds. Take cell you're interested, drug library or individual compound, run the whole thing and query the connectivity map with it. So different sequencing technology, same common fund resource. And what I won't take you through in the interest of time is how that works. And the answer is yes, you can. So you can actually query with data generated in your lab or in this program at really low cost, the CMAP. And from that, you can pull out now measures of similarity. And once again, you'll see, although in some mechanistic sense, it's a superficial comparison, but you'll see once again this sort of overlap with this blocking where compounds of similar have similar activities across the thousands of measurements made in CMAP or repeated in the lab now, and their activities. And whether that biological activity relates to the chemistry is, of course, a critical open question. And then ultimately, the question is, can you take this type of information and derive a mechanistic signature related to a dark kinase? And so the idea is you have a gene signature on which you've drawn a drug query. And I'm just showing you a couple of sort of arrows here that where you switch over from a compound on the one hand to a gene on the other based on RNAi, or in the more contemporary data sets, CRISPR. And that remains to be seen how well that would work, but the idea is then to map out for all the dark kinases what in biology space rather than structure space is the closest chemical hit. And last thing to show you about that is, in anything to do with small molecules, one of the most critical things is dose response. And it seems sort of obvious that you should get an increasing intensity of a signature with dose response. And what you can see here with the L1000 assay, as it exists in the Broad's connectivity map, there is no dose response relationship between the magnitude of a signal measured using RNA sequencing, or in that case, L1000, and the concentration of the drug. And now using these more modern sequencing methods, you can get that. So now you can see the dose response correlation is very, very nice. And that it's not something that you typically think about in gene space, but is absolutely critical in compound space. So we'll be able to relate the details of the dose response in this biology space back to the details of the dose response in chemistry space. So what are next steps in this as I finish off the talk? The first is, there's quite a lot of analysis to be done in that commercial kinome scan data. We're really hoping that Eurofins will collaborate with us on this. There are a fair number of internal consistencies, which is sort of remarkable for a data set that's so widely used commercially. We're trying to get the two different ways that I showed you, one mass spec, one commercial, to be similar. They're not similar. They only overlap by 70%, which is sort of remarkable. There will be then in vitro kinase assays that are developed by other members of Gary's team. And then we're trying to then eventually rationalize the polypharmacology and use that to drive new discovery. I think in light of the talk you're about to hear, it'll be interesting to see whether we can also use it in this fragment-based discovery space. So let me just thank the people who've done that, the active lab members on the left. I won't call them out individually. People who've left on the right. There's a collaboration with Nathaniel and Fleur, also with my close colleague Mohammed Al-Qureshi on the deep learning, and funded by the Common Fund. Gary is the PI of the effort. Thank you very much.
Okay, thanks, Peter. Really appreciate it. Great talk. Really an interesting approach to understand the interaction of these small molecules with the kinome and mass. So we're going to jump to Jarrod right away, and we'll take questions at the end of both talks. So Jarrod, you're on.
Yeah, okay, great, thanks. Hi, everyone. Hope you can see my slides okay. Let me go into the presentation mode. Great, okay. Yeah, thanks for the invitation. Nice to be here again and present to the IDG group. Sorry. Bear with me a second. A little technical difficulty getting the slides to go forward. Yeah, sorry. Quick COI statement that I have to show you. And today I'm going to talk really thematically in many ways related to Peter's talk. So we've proposed to identify some chemical starting points for the dark kinases by screening electrophilic fragment libraries. And, many of you have probably seen many papers coming out recently. This area or this approach seems to be gaining good momentum in the field. And in our case, we have a small compound library, about 300 or so electrophilic fragments, courtesy of Nathaniel Gray, before he left for the West Coast. So it's kind of funny. It's similar to what Peter described. So we have a similar size library, except instead of being clinical grade compounds or selective inhibitors, these are all small fragments, sort of 200 to 300 or so molecular weight. And you see the distribution of warheads here, primarily acrylamides, a few chloroacetamides. And we envisioned, with respect to the dark kinases, sort of two different approaches in which we might be able to use chemical proteomics to identify new binders to the dark kinases. So over here on the right, basically, thanks, Peter, you've sort of described all of the stuff around MIBs perfectly for me, so no explanation there. So this is essentially very similar to what similar approach to what you heard Peter describe, using MIBs as kind of activity-based probes, and then setting up competitive binding assays and reading out sort of differential kinase capture by mass spectrometry. So that was sort of the approach we envisioned on the right, and this is going to allow you to identify fragments that might bind and be competitive within the active site of the kinase. And then thinking, as I've been to a few of the IDG meetings over the years about some of the other resources that are available within the consortium, Ben Major, for example, has produced in his lab cell line reagents where they are stably expressing tagged versions of many of the dark kinases. And so we thought this might be an interesting alternative approach where you could use the affinity handle to do the kinase enrichment. And in that case, you could find fragments that might bind sort of agnostically around the kinase, sites that are outside the ATP binding pocket. And this is, the goal here would be to find perhaps new pockets on the kinases, maybe docking sites for an eventual development of an eventual degrader moiety, things of this nature. So we kind of envisioned two different approaches, and we'd done various work in the past that we thought we might be able to leverage to kind of help us along here. I'll tell you first about a little bit of the effort on the left-hand side here, and sort of in the spirit of warts and all, I'll tell you that we really haven't gotten a huge amount done on this arm. Most of our work has been in the other arm. Here on the left, we're showing sort of the cartoon workflow, if you will, of how we envisioned this experiment going. So we would take cells that express a flag tagged kinase, dark kinase of interest, make extracts, incubate these with one of a pool rather of compounds from our electrophilic fragment library, and then be able to IP the flag tag kinase, digest, encode with TMT multiplex labels, exactly as Peter was describing in his talk, do MSMS, and then we might expect to find, here we're identifying the fragment modified cysteine. I should mention the library are cysteine directed fragments, electrophiles rather. And so we would be directly identifying the peptide harboring a particular fragment on a cysteine, and then we'd have a validation step. So each of the pools is comprised of 5 individual fragments, and so then we do sort of fragment deconvolution, if you will, and sort of read this out. For example, see if we can find dose response labeling in a particular cysteine. So to get started here, we did a little bit of meta-analysis, if you will, comparing some published data along with what Ben reported in his sort of inventory at the time anyway. So we ended up with about 20 or so of these, plus we had a few on hand in our lab from previous studies in different cell lines. And to date, really what we've been trying to do is triage these. As you can imagine, expression levels, even for exogenously induced kinases, are going to be different across the cell lines. And so we've been trying to understand if we can just rapidly correlate expression level with percent sequence coverage for the different kinases, because as you can imagine, if you're having difficulty getting good sequence coverage with reasonable amount of input material, it's going to be hard to robustly identify fragments that might be binding to cysteines. But in a few cases, we're up in the neighborhood of 70% sequence coverage. So I think this will be a viable alternative, a viable pathway for us to find some potential binding at some of these distal cysteines. And I'll spend, sorry, my computer's fighting me here a little bit. So I'll spend most of my time here talking about our progress in this arm. Again, the idea of using the multi-inhibitor beads as kind of an activity probe, and then set up competitive binding assay for the dark kinase, use the MIBs to do biochemical enrichment, and then read out differential binding or differential recovery of the kinases by LC-MSMS. So we got started with these MIBs in sort of a traditional kind of old-fashioned, if you will, shotgun style acquisition. So, in a couple hours acquisition, we're able to monitor about 200 or so kinases, and 44 of these are among the dark kinases. And shout out to Gary and Mike East for kindly providing the reagents and giving us some protocols to get started. That's much appreciated here. And so over time, this is kind of the workflow that we've set up. And so again, the fragments are arrayed in pools of 5. And that's actually, we have a pretty strong effort in, robust effort in screening purified proteins, actually. And that's why we've arrayed these fragments in pools of 5, but it works as well for this effort with the MIBs. So again, we create protein extracts. We incubate with control or pools of fragments. In this case, we're doing then the MIBs enrichment digest. We're encoding with the TMT 6-plex reagents here. You heard Peter mention the 16-plex or 18-plex. They come in different flavors in that respect. And so our idea here is then basically when you have fragments that are binding a particular kinase, you'll see that represented as a reduction in signal here. And so in this way, we can identify hit pools, if you will, for a particular kinase. And then there's a second step where we would take the 5 fragments within a given hit pool, incubate those individually in extracts. And by this, in this way, we would think about sort of doing fragment deconvolution to understand which specific fragment is binding to the kinase. And so we have about 300 fragments total. We're screening them in pools of 5. I think we have about 10 of these experiments to go, but we've done enough now that we can start to reach in to the data and see if we can find at least some interesting trends. So we have control compounds in here. For example, this is this multi-targeted covalent inhibitor SM171. This was developed by Nathaniel, and we co-published with him on this. Actually, Peter's on this paper too, as I remember. And so you can see here as a control, the waterfall plot, you can see we're nicely hitting multiple family members of the MAP kinases, which were well established as to be targeted by this compound. And then other ways to slice the data, we can look within individual pools and find related kinases that appear to be targeted. And similarly, we can look across different pools and see, for example, here's fragments across different pools that are targeting the kinase PCAN3. So hopefully some themes within the data, although the primary screen here is not complete, to give us a sense of optimism here that we're going to find some good chemical leads. And so this is a heat map that kind of shows cumulative data so far. Again, we have about 10 or so of these 10 or so of these reactions yet to acquire. Spans about just under 200 kinases, and we're able to track 60 dark kinases within that set. And here you can see a waterfall plot that is kind of an overlay, I guess you would say, of all the data. And here we're able to track apparent hits. Now, again, we haven't done the fragment deconvolution yet, but apparent hits against 16 different dark kinases. And those are shown here. And the ones in the light blue are sort of predicted at least to have a cysteine within the ATP binding pocket based on some of these papers. But again, we'd have to do the follow-up fragment deconvolution to verify those. So a couple other interesting trends that we can see out of the data. You know, we looked at or we tried to understand the relative abundance of the detected kinases as overlaid on a backdrop shown in blue of kind of relative abundance just in the workhorse cell line here, the HeLa cells that we're using to develop this assay. And, you know, basically you see that the distribution of dark kinases is essentially the same as the set of all kinases that we detect here. And a zoom in here of the TMT reporter ions for these individual guys. So here you can see, you know, this is pool number 3 that appears to have a fragment that's reacting with the active site in CDK12, and hence, you know, disrupting its interaction with the MIBs as the activity probe. And then similarly for this MAP kinase guy. All right. So this slide was intended actually to be part of the justification for why we also took the second arm approach with these affinity tag kinases, and that it's really shown here on the left. If we look, of course, at the sequence coverage we get across the dark kinases, if we wanted to find sites outside the ATP binding pocket, you see that, you know, we struggle a little bit with sequence coverage on some of the lower expression kinases, and that was part of our motivation to go after these affinity tag kinases to get get an ability to do strong biochemical enrichment of those guys to increase our sequence coverage. And so, as I mentioned earlier, that work is kind of still in the early stages, but percolating along. Okay, so I'll take a minute here at the end to just mention a couple other newer approaches that we think will move the needle a little bit for us in terms of improving the assay that we've developed. So one is we published here with Gary and others in the IDG consortium, sort of our version of targeted mass spectrometry, something we called PRM Live. And this is referring back to Peter's talk. This is sort of a notion of doing the targeted analysis where the computational predictions are all now focused on retention time of the peptides of interest and sort of adjusting those in real time so that we don't require stable isotope labels as internal controls. As Peter mentioned, sometimes those can be kind of tricky to get a hold of and whatnot. So anyway, we published this paper really independently of our work in the dark kinases, but even in our initial look, we were covering 38 dark kinases in this assay, including 10 of the 16 that we see hits for so far in our primary screen. So the libraries, are only getting bigger. Actually, the fragment library we have is, I would say by all measures, pretty modest in size, actually. And so to really go after these using this kind of approach with sort of the kinative style approach, I guess you would call it, which of course has some advantages because you're looking at binding of endogenous kinases, in their native environment and all that. So anyway, I think we need to sort of continue to push towards ways to improve the throughput and multiplexing. Peter mentioned in his talk, going towards this TMT 16-plex reagent approach, for example. So we're pretty optimistic about bringing this technology to bear. And the other one that I'll finish with is even more recent. This work is under review right now. This is based on a collaboration between my lab and Brooker and 908 Devices, and 908 makes this really slick electrophoresis. It's kind of essentially the size of a microscope chip. You can see that shown schematically here. And basically it has a small channel and a sample and a serpentine separation channel here, and then it ends in the corner with the emitter, and that's how you generate the electrospray for direct integration with the mass spectrometer. And this has been used pretty successfully over the past few years for, purified proteins and other solutions that are, sort of concentrated. But it's not been such a good fit for complex proteome samples. And we've been working with them on a prototype implementation of this, where they've incorporated sort of a miniature LC bed, if you will, reverse phase or solid phase extraction bed here directly on board on the chip. And so this sort of gives you an ability to pre-concentrate samples onto the chip and then get ballistic separations in the electrophoretic mobility channel. And so we've done multiple types of applications with this, but the one that we've got just under review now is sort of what we call warp speed activity-based protein profiling. And you can see, within like a 10-minute acquisition window, you can get reproducible quantification of, over 200 peptides here. So we think this might be a nice new avenue to really increase the throughput. So these acquisitions are done in about 15 minutes. And so we think that's a very nice way to get additional multiplexing in there, accurate quantification to think about tackling some of these larger libraries. And so I think that's just my quick summary slide, still in early stages here, as I mentioned, for looking at binding using these flag tagged reagents. Much more progress here for us in terms of using the MIBS activity probes to find potential hits, potential covalent fragment hits to the ATP binding pocket here. And so we're just about done, as I said, with our primary screen, and then we're preparing to launch our fragment kind of deconvolution So we need to confirm, covalent mechanism of action and, site modification and all that kind of stuff. And hopefully we'll be able to bring some of these new technologies for the high throughput PRM to bear in our work as well. And with that, I'll just put the acknowledgements up. Folks in my lab that have helped with all this work, our collaboration with 908 and Brooker, and then many of the folks in the IDG. It's very much appreciate all their willingness to help us out get some of these assays up off the ground. So thanks very much for listening.